This is the Diversity of Life Simple Organisms lab demo part for the online Bio 113 section. So we're looking today at prokaryotic cells and eukaryotic cells. I just wanted to remind you again, the prokaryotic cells, those were the domains of bacteria and archaea, single-celled organisms. Keep in mind the sizes we're talking about, 1 to 10 microns, micrometers. DNA is usually circular. They have very few cellular structures, and they divide by binary fission, which means they divide really fast. They always double too, right? 1 makes 2, 2 makes 4. This is your first example of exponential growth. And then we have eukaryotic cells. They're about 10 to 100 times bigger than prokaryotic cells. Eukaryotic is eu means good, and karyotic, that's from karyon. That's the Greek word for nut. So those are the ones with a good nut. Those are the ones with the nucleus on the inside. They're bigger, and because of that, they can't rely on diffusion solely. There's a prokaryote over here. Those are the cells in plant and animal cells. Those are the cells in multicellular organisms. They're between 10 and 100 microns in size. Their DNA is in the nucleus. They have chromosomes. They have organelles to deal with that diffusion problem. And they divide by mitosis and meiosis. I wanted to remind you of the bacteria. What we're looking at here is we're looking at basic shapes. The shapes a lot of times inform the names. So if I'm asking you to look at Bacillus cereus, we're going to be looking at rods. If I'm asking you to look at Streptococcus pyogenes, coccus, that means it's a small sphere. And because it's a strepto, it's also probably going to be in a chain. Staphylococcus is going to be in a cluster. And spirillum species are going to be a corkscrew looking thing. I spoke in the lecture part of the distinction between gram-positive and gram-negative bacteria and how it relates to their sensitivity to antibiotics. Gram-positive bacteria are sensitive to penicillin, while gram-negative ones are not. And this has to do with the lipid the membrane of gram-positive versus gram-negative bacteria. Gram-positive bacteria, their cell membrane, cell wall, not cell membrane, their cell wall looks like this. It's got a thick peptidoglycan layer. Gram-negative bacteria also have a peptidoglycan layer, but it's much, much thinner. And they actually have a much more, they have lipopolysaccharides on the outer membrane, which actually are a problem with gram-negative bacteria. So gram-positive bacteria are things like your Streptococcus and your Staphylococcus. So those are sensitive to penicillin and associates. These guys here are not sensitive to penicillin because peptidoglycan is not that big of a structural thing for them. But these guys, this lipopolysaccharide is actually a problem for us as our immune system takes these babies apart. This lipopolysaccharide gets released and we react badly to it. Which is why gram-negative bacteria give us things like diarrhea and sepsis and that kind of stuff. So, but the reason why they're called gram-positive and gram-negative has to do with this specific stain that we've got here, which is called the Gram stain. And the way the Gram stain works is you make your bacterial smear. The way you make a bacterial smear is you take a slide, you grab a loop full of bacteria that you spread on the slide from a culture. And then you add crystal violet, which is a purple dye, to it. You wait a minute. And then you add iodine. What iodine does is the crystal vial goes inside of the cell. The iodine goes inside of the cell, so it goes inside of the cell membrane, both of them do. I should give you hang on, pointer options. What do we got here? Laser pointer, pen. So the crystal violet goes inside of the cell all the way in, and it goes inside of the cell for both of them. The iodine also goes in, and what the iodine does is it makes a complex that's actually larger on the inside. And again, it goes in here too, and it makes a complex that's larger than it was before. And then you come with an alcohol wash. What the alcohol wash does, it is washes this whole thing back out. Well, however, because you have this big, huge membrane here, this peptidoglycan layer, it keeps the alcohol from washing it out. Whereas here, because that peptidoglycan layer is very small right here, the alcohol can wash this out, so this goes away. So after the alcohol wash, the gram-negative bacteria have lost their color. And then we're coming after it with saffron and with a red stain. That's our counter stain. And the counter stain, because the dark blue, the purple dark blue is so dark, you can't see the pink. The pink actually goes into both of them, but you can't see the pink because it's so much lighter than that dark color. And so by having this stain here like this, we know that these guys, those are the gram-positive ones. Those have the peptidoglycan layer. And these guys are the gram-negative ones. And that's how the gram stain works. And the gram stain is so useful because it also tells you something about the sensitivity to antibiotics. And here's one more time if you wanted to look at. So this is what we're talking about here. Is we're talking about the cell wall peptidoglycan synthesis. So you've got some cells have this peptidoglycan wall, and some of them have this lipopolysaccharide outer membrane in gram-negative bacteria. And the peptidoglycan wall, that's the gram-positive bacteria. A lot of your activities are going to be available on the Google site today. So one of the things I want you to do is I want you to actually try to look at one of those microscopes, or possibly both of them, because it's a very different experience to try to look for bacteria under a microscope than it is to just look at them, like I'm showing you here, bacterial shapes. Because on the microscope, the first thing you're going to have to figure out is, what am I even looking for? And so let me show you on this microscope here. So this one has the bacterial capsule as an option. And so move it. I like looking at the checklist over here. Come on, how are we gonna turn this thing on? Here we go. All right, I'm gonna switch views, adjust the eyepiece. so here's the first thing you see. And when students start looking at this, they tend to think that this big thing here is the image they're looking for. No, no, no. That's just the edge of your stain. Let me show you. That's just the edge of your stain. That's where the color is most intense. And what you really want is you want to look somewhere, in here or so. Those little dots that you can't barely see, those are the dots you want. So let's go and fine focus this thing to the point that it's happy. There we go. Switch the view, and then you twist it to go to the next objective. Switch view again. I think it's still centered enough. Center the specimen again. Oops. Fine focus until it's happy. No, that's dirt. No, that's the wrong direction. There we go. Now it's happy. And this is a capsule stain, so it's not a stain that we've done, which means it shows it's actually a negative stain. So it shows your bacteria are clear against a dark background. So see, the bacteria are the little light colored dots, actually. So let's get to 1,000. And now let's fine focus it just till it's happy with us. Not that way. There we go. So the capsule stain, what it does, it shows a negative, so the color is the background. And this one is a good image here. This is, so on the inside is the bacterium, and this is the capsule part. So these pieces that are barely visible, that's what you're looking for. So keep in mind, you're at 1,000x here, right? And you're barely going, am I really seeing what I'm supposed to be seeing? And that's the experience I want you to have. That's the experience you don't have unless you're trying to look at a microscope. Similarly, this other microscope here, and I just went for explore. And open the slide box here. And there are a number of bacteria slides. And there's an acid-fast mix, that there's an endospore stain, and then there's a gram stain mix. I'm going to click on the endospore stain real quick. And we're at 4x. So this one allows you to course. And similar to the other one, this one just allows you to adjust the light, but you're just, so those little, basically invisible things there, that's what you're looking for. So let's go to 10x. Now you just want to look at fine focus. Oh, no, it still allows you to do coarse focus. This didn't do much here. A little more light. 40X. And remember, this is an endospore stain, so this only looks at the blue things inside of the bacteria, right? And then when you go to the 1000X, it'll ask you to add immersion oil. And then your fine focus, you need more light. You need your fine focus adjust. And what your spores are is probably these little purple things. I'm not even sure, actually. Oops. Yeah, see, it doesn't want us to use that knob at all. So this really is a pretty decent example of this is what a crappy stain looks like. And you're like, what am I even looking at here? So, but this is what, this is the kind of experience I want you to have to make sure that you understand that just because it's under a microscope, it's actually a skill. And so as I'm thinking about this, it's probably these little purple things that are going to be your positives. I don't think the blue ones matter. But I'm not 100% sure. On that front, well, we'll get to that in a second. Okay, back to the... So now that you have a newfound appreciation for what we're trying for here, right? You can watch here how to make a stain. This is a basic stain, and in the lab, we would normally do a basic stain. These are bacterial shapes, and it also talks about how the clusters and the chains come about, because this is not, they don't arrange like that on purpose. Well, not on purpose per se, but it's something about the planes of division that cause it to be one way or the other. So the first thing that I want you to do is to, on page 8, your first submission, there's a lot of sketching going on this time. So you should sketch what you see after you've stained Bacillus cereus. And this is an example of actually Bacillus cereus over here on the left. So it's a rod-shaped bacterium that has spores on the inside. See those little purple dots on the inside there? Those are actually spores. And so the bacterium is the whole big thing, and the little purple dots are the spores. So if you have a Bacillus cereus, what's the shape of the bacterium? You should be able to answer that. I'm going to give you a quiz for the questions, but you'll still have to submit your lab notebook for the drawings. But go ahead and submit the quiz for the questions, because that way you can get credit for that. Do these cells, so you're asked, do these cells grow singly or in chains? And that's pretty obvious. What details of cell structures you can make out? I just mentioned that to you, right? Then the next, so microscope, usually what we have in microscope 1 is this one here, the first one. Then this, so you're supposed to draw this, right? So draw the shape of the what's under the microscope. Draw the rod-shaped bacterium, Bacillus shape, and then draw the Spirillum-shaped bacterium. Then the next stain is a Gram stain, and over here on the right is an example of a Gram stain. And you see the two different types of bacteria there. You see the round, the purple ones that are the Gram positives, and the gram negatives that are rod. Why is the gram stain reaction performed, and what's the major cellular difference? You should know both of those now. Now, oscillatoria, here's an example. There's a picture of oscillatoria. When we see those in the lab, we usually end up they're like a big clump, and people grab that big clump and then just go for the darkest possible color. But the reality is you have to go, you go in focus on the darkest color, but then you go off to the side to try to see the detail. Because the detail is that each one of those individuals is actually an organism. This is an organism, and this is an organism, and this is an organism. Each one of them is a cell. These are just floaty oscillatoria, so nothing major. And then there's a question about the color. The next one you're supposed to sketch and label is euglena. And there's a couple of questions associated with this. The way this one shows euglena in motion, and what you normally see in the lab is something a little bit like this. This is usually what we see. Hang on, can I enlarge this? This is the kind of thing you see. Now we have something that we can give them to slow them down, which is more or less what we've got here, where they're basically getting stuck, and then you can watch them more closely. But they're really way more fun to just watch them float around and stuff. And on this video, you can actually see, if you're watching it all the way through, you can actually see the flagellum, and you can see it a little bit here. But there's a couple of other ones where you can see it better. So, but we'll exit this back out. We'll stop this. And then there's Volvox. So there's another drawing where you want to label the cell, the matrix, the flagella, and the daughter colonies. If you want to, you can find a picture online of one, or you can grab one of these pictures and label the different structures. And then we get to yeast. This comes from a website from a winery, actually. So there's Saccharomyces cerevisiae at 100x. There's a question about it at 10x and 100x. So this is the 10x because it's a total magnification of 100, and this is the 400x, which is a 40x total magnification of 400. So this is what you see. So just describe what you see. And then the next one is mushrooms. So we've got a mushroom with its stalk and its gills. And then that's a bracket fungus. And there's a Zygomyces, Penicillium, and Aspergillus. For Zygomyces, Penicillium, and Aspergillus, what I'd like for you to do is actually find some online pictures of like them under a microscope, and ideally submit those, because you really want to see the sporangia and the spores and the mycelium and what that looks like. And then last but not least, we've got examples of lichen, the crustose type, the foliose type, and the fruticose type.